Biomolecules are the all organic molecules which are present inside the living system or a living cell are called as biomolecules. We know that if you take any living organism, it is made up of various types of elements which may be either organic or inorganic. So all the organic molecules which are present inside a living cell are called as biomolecules. Why we are saying biomolecules is, here bio means living. That means the organic molecules which are present inside a living system or inside a living cell. Okay, here these biomolecules are made up of various types of elements like carbon, hydrogen and oxygen. Apart from this carbon, hydrogen and oxygen, they are also made up of the other elements like nitrogen. So these are the four major elements by which the biomolecules are made up of. So let's come to the other side of these systems, that is when you come to non-living systems, like what is a composition that is present in the non-living system of the non-living organisms, or let's take an example of earth crust. So earth crust is also made up of the same type of elements by which the living system or living cells are made up of. Then what is the difference between the non-living systems like earth crust and the living systems like living organisms? The only difference is, okay, the proportion of the carbon and hydrogen, that is the abundance of carbon and hydrogen in the living cells when compared to the non-living systems. But in other part of other part of this one, we can say that the elements which are present in the non-living systems are present in the living systems also. Then how can we know that which type of elements are present in the living systems? So that can be done by a process called as elemental analysis. Then by elemental analysis, we can know the various types of elements which are present inside a cell or inside a living system. And after knowing about the type of elements, we have to know about which type of compound is present. So then after knowing about the type of compound, what is the molecular formula, which type of elements are present in that molecule have to be known. All that is done by various types of process, that is called as chemical analysis, analytical analysis, and also by doing the ash test or ash analysis test. So how can we know that presence of various types of elements or compounds in a part of any organism is? So first we have to dry the part of the plant, so that when we dry the part of the plant, all the water present in the plant body will be left out. And after drying up, we have to burn it. After burning that, we get the ash, and when we analyze the ash or when we test the presence of various types of elements in the ash, we come to know that various types of elements and also compounds which are present in that plant body can be known. So this is what we say that analysis or the ash analysis, which gives the information about the presence of various types of elements and also the compounds which are present in the living cell or any living system. Then here all the macromolecules or the micromolecules, we can say that the molecules which are present or the organic molecules which are present inside a living organism are called as a biomolecules, and these biomolecules are majorly made up of carbon, hydrogen and oxygen. And here which type of organic molecules are present in a living system? A living system may be made up of various types of organic molecules, like it may be proteins, amino acids, polysaccharides, sugars, nucleic acids like lipids, fats, fatty acids, like various types of organic molecules are present. And all these organic molecules are coordinated somewhere with other type of organic molecules, such that the process or metabolic process is going on inside a plant body. Likewise, knowing about various types of organic molecules is studied under biomolecules. Now let us discuss about the questions, and when we are discussing the questions, let us know something about the properties, the characters and functions related to various types of biomolecules which are present inside a plant cell and also in the animal cells. Question number 1. The acid used for grinding the vegetables or piece of liver to analyze the organic compounds is options: 1. Tricarboxylic acid, 2. Acetic acid, 3. Trichloroacetic acid, 4. Malic acid. Okay, here grinding the vegetables or a piece of liver, this is a technique or the process which is used in the in the process of knowing about the type of organic compounds or organic chemicals which are present in the living cells. So for this process, what we do is we take a piece of vegetable or a piece of liver of any animal, and that have to be done with crushed with the help of pestle and mortar by using a chemical called as trichloroacetic acid, that is Cl3CCOOH. This is a trichloroacetic acid, which is a chemical used for analyzing or knowing about the presence of various types of organic compounds present in a plant cell or a part of vegetable or a part of liver that is in the animal cell. By using this trichloroacetic acid, a slurry is prepared, and that slurry is filtered, and we get two parts. One is called as a filtrate and the retentant, that is acid soluble fraction and the acid insoluble fraction. In the acid insoluble fraction, we can see the presence of that is both in the acid soluble and acid insoluble fractions. Whatever we are getting, we can know the presence of various types of organic compounds present in that living organism or in that part of that plant body or the animal body. So question number 1, correct answer is 3. Question number 2, all the carbon containing molecules present in the living tissues are commonly called. Options: 1. Heterocyclic molecules, 2. Heteropolymer molecules, 3. Biomolecules, 4. Dicyclic and heterocyclic molecules. Okay, let's come to the first option, heterocyclic molecules. Heterocyclic molecules means the molecules which are having cyclic nature, and those two cyclic nature are different, that is they contain at least two different types of cyclic compounds in that one. Then second one is heteropolymer molecule. So the word polymer means it's an giant molecule which is made up of repeated units, and each unit is differing from one another. Such a polymer is called as a heteropolymer molecule. Then third one is biomolecules. All the molecules which are present in a living system or in a living cell are called as biomolecules, and these molecules are nothing but organic in their nature, and they are mainly made up of carbon, hydrogen and oxygen. Then fourth one, dicyclic and heterocyclic molecules. Dicyclic means a compound which is made up of two cyclic structures, and those two cyclic structures are differing from each other. So we call dicyclic and heterocyclic compounds. Okay, when you come to the conclusion part, so we can say that all carbon containing components which are present inside a living cell are called as biomolecules. Biomolecules are nothing but the organic molecules which are mainly made up of carbon. Apart from that one, hydrogen, oxygen is also there, and this type of organic molecules are present inside a living cell. Question number 2, the correct answer is 3. Question number 3, the major percentage weight of element present in the human body among all other elements compared to earth crust is that of. Options: 1. Nitrogen, 2. Carbon, 3. Oxygen, 4. Calcium. Okay, here when you are talking about the type of elements present in the human body, we know that the human body contains various types of elements, but the composition wise, if you take the percentage wise, it is differing from one another. Out of the given options, we can say that oxygen is the one which is of 65% by weight. That means if you say that all the elements which are present in a human body are of 100%, out of that 100%, oxygen itself is occupying about 65%. So that major percentage weight of the element present in the human body is of oxygen, which is of 65%. So question number 3, the correct answer is 3. Question number 4, the primary structure of protein is formed by. Options: 1. Phosphodiester bonds, 2. Peptide bonds, 3. Glycosidic bonds, 4. Hydrogen bonds. Okay, what is a protein? Protein is an polymer of amino acids, or we can say amino acids are the building blocks of protein. And here in a protein, many amino acids are there. And one more thing, that basically each protein is made up of 20 types of amino acids. 20 types of amino acids. And these 20 types of amino acids may be repeated many times. And one more thing, that it is not compulsory that all proteins should be made up of the 20 types of amino acids, but the maximum types of amino acids that are present in a protein are 20 types, and they may be repeated n number of times. N number of times is many number of times, and if similar type of amino acids are there in one protein, we call it as a homopolymer. If different types are there, we call it as a heteropolymer molecule. And how does the protein is forming into chain like structure means, it is by a special type of we can say strong bond, that is called as a peptide bond. And how does a peptide bond is formed means, for example, if you take some of the one part of the example here, that this is a basic molecular structure of a amino acid. Consider this is one amino acid and one more amino acid is there here. In each amino acid there are three functional groups. One is called as carboxyl group, amino group, and R represents the alkyl group. Okay. So likewise here, the first, if you consider this as first amino acid and the second amino acid. During the formation of bonding, what happens is the bond is always formed between the COOH of earlier amino acid to the NH2 group of the next amino acid. When the bond is formed, one water molecule is lost from this one, and the type of bond that is formed is called as peptide bond. In the same way, the second amino acid is binded to the third amino acid, in which the COOH group of second amino acid is binded to the NH2 group of the third amino acid. So likewise, a chain of amino acids is formed with the help of an peptide bond. And now the protein chain which is formed will be a straight one, without any helices or without any coilings. And protein chain which is not having any helices or coilings is called as a primary structure. And one more thing, that proteins not only form the primary structure, they also form secondary structure, tertiary structure, and the quaternary structure. So let us point out what is the difference between primary, secondary, and tertiary here itself. Primary means the protein chain without any helices is called as primary structure. Secondary means it forms a helical structure, that is a secondary. Tertiary means the coiling is overlapping one above the other. And quaternary means chains of proteins will be overlapping one above the other. So likewise, we can see the difference between the primary, secondary, tertiary, and the quaternary structures. And in the primary structure, the structure is stabilized by the formation of the peptide bonds. So question number 4, the correct answer is 2. Question number 5. The simplest and least carbon containing amino acid is options 1 glycine, 2 serine, 3 lysine, 4 alanine. Okay. The given options: 1 glycine. Glycine is a amino acid which is having 2 carbons in it, that is C2H5O2N. This is a molecular formula of glycine molecule. Second one is serine. Serine C3H7O3N. It contains 3 carbons in it. Okay, second one is serine. The molecular formula is C3H7O3N, which contains 3 carbons in it. The third one is lysine. C6H14O2N2. So this is a amino acid which contains 6 carbons in it. Then fourth one is alanine. C3H7O2N. It also contains 3 carbons as like in serine molecules. So here these are the 4 compounds that have been given in the 4 options, and these are the molecular formulas, that is glycine C2H5O2N, serine C3H7O3N, lysine C6H4H14O2N2, and alanine C3H7O2N. And out of these 4, glycine is the molecule which is having least number of carbons, that is 2 carbons are present, and the one which is having the least carbons and also the simplest molecular structure. So question number 5, the correct answer is 1. Question number 6, the ratio of hydrogen atoms present in the glucose and ribose sugar respectively. Options are 1 6:5, 2 1:1, 3 5:6, and 4 2:1. Okay, here the 2 molecules that have been given in the question are glucose molecule. So glucose molecule is an monosaccharide which contains 6 carbons in it, that is a C6H12O6. So this is a molecular formula of glucose molecule, and the second one is ribose sugar. Ribose sugar is a pentose sugar which contains 5 carbons in it, which is a part of RNA molecule or the nucleotide of RNA molecule C5H10O5. This is a molecular formula of ribose sugar. So let's come to the ratio of hydrogen atoms present in glucose and ribose sugar. In glucose molecule, the number of hydrogens are 12, whereas in ribose sugar, the number of hydrogens are 10. When we simplify this 12:10, it comes to 6:5. So the ratio of hydrogen atoms present in glucose and ribose sugar respectively are 6:5. Question number 6, the correct answer is 1. Question number 7, the chemical and physical properties of amino acids depend on. Options 1 COOH, that is carboxyl group, NH2, that is amino group, and R group. R group means alkyl group. Second option carboxyl group and amino group only. Third one COO minus, that is ester form of the carboxyl group, and NH plus, that is ionic form of amino group. Fourth one carboxyl group. So here in the 7th question 4 options have been given. Out of the 4 options we have to select that the chemical and physical properties of amino acids are based on. Actually, if you take an amino acid, this is a molecular organization that is present in amino acid, where NH2 group, alkyl group, and the hydrogen. So this is a basic formula, basic formulation or the structure of the amino acid. And here this R is an alkyl group which may be replaced by hydrogen as in the case of glycine, or it may be replaced by methyl group in the case of alanine, or it may be the CH2OH group as like in serine. And here the physical and chemical properties of any amino acid is based on all the functional groups which are present on the carbon. So that in the given options we can say that carboxyl, NH2, that is amino group, and the alkyl groups. Question number 7, the correct answer is 1. Question number 8, identify the Zwitter ionic form of the amino acid in the molecular structures shown below. A, so that it is given as a CH, R, COOH, and NH2, NH3 plus, and B is C, COOH, NH2, and R. This is the second option that is given, and C is C, COO minus, and NH2 plus. So these are the 3 options that have been given, A, B, and C. From this we have to identify which one is a Zwitter ionic form of an amino acid. So first let us come to one point, that what is a Zwitter ionic form. So Zwitter ionic form is a one which contains both a negative charge and also positive charge on the amino acid structure. So out of the given options, let's take here the first one. Here COOH is there and NH3 plus is there, that is the cationic form that is positive charge is present, only positive charge is present, whereas the second one, so in the B it is having only negative charge on it, that is COO minus, but there is no positive charge. Whereas the third one, COO minus, that is negative charge is there, and also NH2 plus, that is positive charge is there. Both negative charge and positive charge, both of them are present, so we call this C as an Zwitter ionic form. So question number 8, the correct answer is C, that is option number 3. Question number 9, the correct set of components found in nucleotide are options 1, homocyclic compound, monosaccharide sugar and phosphoric acid. 2, homocyclic compound, pentasaccharide and phosphate. 3, heterocyclic compound, pentose sugar and phosphoric acid. 4, heterocyclic compound, pentasaccharide and phosphoric acid. Okay, here first let us come to the point that what is a nucleotide. So nucleotide is a basic unit of a nucleic acid, or we can say that nucleic acids called as DNA and RNA, they are made up of the polymer of nucleotides. That means many nucleotides are present in them. And what is the composition of each nucleotide? Each nucleotide is made up of 3 structures. One is called as nitrogen base. The second one is called as pentose sugar. And third one is called as phosphate group. These are the 3 things by which the nucleotide is made up of. And what are these nitrogen bases? Nitrogen bases are nothing but the purines and pyrimidines, that is adenine, guanine, cytosine, uracil and thymine. Then pentose sugar. Here there are sugars which are made up of 5 carbons, and there are 2 types of sugars are there. One is ribose sugar and the deoxyribose sugar. Ribose sugar is present in RNA molecule, whereas deoxyribose sugar is present in DNA molecule. The basic difference between ribose sugars and the deoxyribose sugar is deoxy, that is the sugar molecule which is having 1 oxygen less than that of the ribose sugar. Then phosphate molecule. Phosphate molecule is a PO4 minus, or we can say the phosphoric acid. So these are the 3 components which are the part of the nucleotide molecule. Now let us come to the conclusion part, that is nitrogen base. Nitrogen base is an heterocyclic compound. What is the meaning of heterocyclic compound? Means it is a cyclic molecule which is made up of different types of elements in it. That is, a nitrogen base is a ring compound which is made up of C and N, that is carbon and nitrogen. So here it is made up of CN rings. So we call it as an heterocyclic compound. Then pentose sugar, so that is ribose sugar or the deoxyribose sugar. Then third one is phosphate in the form of phosphoric acid. In the form of phosphoric acid. So from the given options we can say that nucleotide is an structure which is made up of the heterocyclic compound, that is CN ring. Is an heterocyclic compound. And pentose sugar along with a phosphoric acid. So question number 9, the correct answer is 3. Question number 10, the heteropolymer biomacromolecules are options 1, all polysaccharides, 2, all lipids, 3, all proteins, 4, cellulose. In the given options, all the molecules that have been given here, polysaccharides, proteins, lipids and cellulose, all of them are biomolecules. That means they are made up of carbon, hydrogen and oxygen. Basically we say carbon compounds. And here what is the meaning of heteropolymer? Here we have to know something about the heteropolymer molecule and a homopolymer molecule. Polymer means it's a molecule which is made up of repeated units. And if the molecule or the structure which is repeated many times in that structure, if it is of a same type, we call it as a homopolymer. And heteropolymer means the repeated units are of different types, so we call it as a heteropolymer. If we take the options here, polysaccharides. Polysaccharides are giant molecules which are made up of many monosaccharides, and this is an homopolymer biomolecule. Then coming to the lipids. Lipids are also homopolymer molecules, whereas proteins are the heteropolymer biomacromolecules, and cellulose is an homopolymer. So here from the given options we can say that question number 10, the correct answer is 3. Question number 11, the number of carbon atoms present in the arachidonic acid. Options 1, 16, 2, 19, 3, 20, 4, 10. Okay, here arachidonic acid is a type of fat. It's a type of fat. So here when you are talking about the fats are the organic molecules which are of two types. One is fatty acids and glycerols. Fatty acids and glycerols. Fatty acids and glycerols are independently present, and these are the molecules which may be either in the saturated form or in the unsaturated form. When you are saying in the saturated form means here they have single bonds between the carbons, like, and unsaturated means which have double bonds between them. When we are saying double bonds are unsaturated character, so they are ready to form a new bond with a new element or the molecule. So that type of nature is there with the fats. And here the number of carbons present in various types of fats are ranging up to maximum of 20, ranging up to 20. And the best example, the one which is having the highest number of carbons is arachidonic acid, that is carbon number is 20. Question number 11, the correct answer is 3. Question number 12, choose the correct statement with reference to trihydroxypropane. Options 1, it is a type of simple saturated lipid, glycerol. Second, it is a type of complex saturated lipid, glycerol. 3, it is a type of complex unsaturated lipid, glycerol. 4, it is a type of simple unsaturated fatty acid. Okay, trihydroxypropane. Trihydroxypropane is a simple saturated lipid which is called as glycerol. It's a simple saturated lipid. Question number 12, the correct answer is 1. Question number 13, the ratio of dicyclic and heterocyclic ringed structures and monocyclic and heterocyclic ringed structures present in the nucleic acids is. Options, first one 1:1, second 2:3, third 1:2, and fourth 1:3. So let's come to the first one, that is dicyclic and heterocyclic. Dicyclic and heterocyclic ringed structures. Then monocyclic along with the heterocyclic ringed structures. In this one, we are talking about the nucleic acids. Nucleic acids are made up of nucleotides, or we can say it's a polymer of nucleotides, and each nucleotide contains a nitrogen base, a pentose sugar, and a phosphate group. And what are the type of nitrogen bases which are present here? There are two types of nitrogen bases are present. One is called as purines group and other is called as a pyrimidines group. When we are talking about the purines group, as we know that they are made up of CN rings. Those are adenine and guanine. They are made up of CN rings, and the basic structure is like this. This is one ring structure which is made up of carbon and hydrogens, and the other ring structure, it will be like this. So the two rings are present. which are different from each other. So we call it as a dicyclic. And why we are calling it as heterocyclic means it is made up of carbon and also nitrogen. The ring is made up of carbon and also nitrogen. So we call it as a heterocyclic compound. So how many nitrogen bases or how many structures are having this dicyclic and heterocyclic are adenine and guanine. So that the ratio is coming to 2. Then coming to pyrimidines. Pyrimidines are thymine, cytosine and uracil. Thymine and cytosine are present in DNA, whereas thymine is replaced by uracil in RNA molecule. That is, RNA contains cytosine and uracil. And these pyrimidines are having CN rings as like that of purines, but the difference is it have only one cyclic structure. That is, CN is present but one cyclic structure. And why we are calling it as heterocyclic means it is made up of two types of elements in it. So heterocyclic. The basic structure appears to be like this. Basic structure appears to be like this. So here how many are of with dicyclic and heterocyclic is, that is 2. And how many are with monocyclic and heterocyclic are, that is 3. So what does this 2 represent means adenine and guanine. What does the 3 represent means thymine, cytosine and also uracil. So the ratio of dicyclic and heterocyclic ringed structures and monocyclic heterocyclic ringed structures in the nucleic acids are 2:3. Question number 13, the correct answer is 2. Question number 14, the building blocks of nucleic acids are options 1 nucleosides, 2 nucleotides, 3 nitrogen bases, 4 monosaccharide, sugar and phosphate. Okay, first options, that is nucleosides. Nucleosides are also the part of nucleic acids, but these are made up of only nitrogen base plus pentose sugar. Then second one is nucleotides. Nucleotides are made up of nitrogen base plus pentose sugar, along with it the phosphate group is present. Third one, nitrogen base. Nitrogen bases are of 5 types already, as we know, that is adenine, guanine, cytosine, thymine and uracil. Then fourth one is monosaccharide, sugar and phosphate. Monosaccharide, sugar and phosphate. Monosaccharide sugar means pentose sugar and a phosphate molecule. We know that nucleic acids, that is DNA and RNA, are the polymers of nucleotides. So in DNA, 2 such strands are there, whereas in RNA, 1 strand of polynucleotides are present. So from the given options, we can say that the building blocks of nucleic acids are nucleotides, which in turn are made up of 1 nitrogen base, 1 pentose sugar and 1 phosphate molecule. Question number 14, the correct answer is 2. Question number 15, abrine and resins are options 1 toxic secondary metabolites, 2 lectin type secondary metabolites, 3 terpenoid secondary metabolites, 4 alkaloid secondary metabolites. Okay, here in the most of the part the word is used is a secondary metabolite. So here we have to know something about 2 things, that is primary metabolites and second one is secondary metabolites. We know that inside a cell or in a living system, there are thousands of or we can say millions of organic molecules, and out of all these organic molecules which are present inside a cell, do we think that all these are having any specific function which is useful for that living organism? Means it may or may not be useful. Based on that character, the organic molecules are categorized into primary metabolites and the secondary metabolites. Primary metabolites means the organic molecule which are having some specific function or identifiable function, or the function that is useful for the organism in which that organic molecule is present. Such type of organic molecules are called as primary metabolites. Then second one is secondary metabolites. Secondary metabolites are those organic molecules which are not much useful for the organism in which they are present. Otherwise, we can say that they does not have identifiable function inside the living system. Such type of organic molecules are called as secondary metabolites. So in the secondary metabolites also there are various types of subgroups are there, like toxic materials are there, terpenoids are present, and the same way the pigments and various types of organic molecules are present. So now let us come to the options that have been given. So abrine and resin are the poisonous materials or which are toxic in nature, and they does not have any identifiable function inside the organism. That's the reason why they are the part of secondary metabolites. So question number 15, the correct answer is 1. Question number 16, the molecular arrangement in nucleotides of nucleic acid is options 1 phosphate, nitrogen base and phosphate, 2 nitrogen base, phosphoric acid and pentose sugar, 3 phosphoric acid, pentose sugar and nitrogen base, 4 pentose sugar, phosphoric acid and nitrogen base. Now let us come to the first one, that is phosphate, nitrogen base and phosphate. In the given option, it shows that nucleotide is having 2 phosphates in it. So this is incorrect. The second one is a nitrogen base, phosphoric acid, along with it the pentose sugar. In this given option, the nitrogen base, phosphoric acid and pentose sugar, the sequence that is given is incorrect. All the 3 are correct which are the part of nucleotide, but the sequence of arrangement of these 3 structures is incorrect. Third one, phosphoric acid, pentose sugar and nitrogen base. Okay, in a molecule or in a nucleotide molecule, if you see the organization, so this is a pentose sugar, so 5 carbons are there, first carbon, second carbon, third carbon, fourth carbon and the fifth carbon, and the first carbon is binded to the nitrogen base. First carbon is binded to the nitrogen base, whereas the fifth carbon is binded to the phosphoric acid, that is phosphate molecule, that is oxygen of the phosphate molecule actually, and one more is there which is binded, third carbon also get binded to one more phosphate molecule. But here the pentose sugar is binded to 2 phosphate molecules, but when you are saying about a nucleotide, one nucleotide contains only 1 phosphate, that is 1 phosphoric acid. If you see the organization, so first the phosphoric acid is coming, the pentose sugar is coming, then the nitrogen base. So the sequence which is given here, it is correct. And the fourth option, pentose sugar, phosphoric acid and nitrogen base, that is also incorrect. So from the given options, question number 16, the correct answer is 3. Question number 17. Identify the correct set of nucleosides of DNA. Options: 1. Adenosine, guanine, cytidine, thymidine. Second option: adenosine, guanosine, thymidine, cytidine. Third option: adenosine, uridine, thymidine, cytidine. Fourth option: adenosine, guanosine, uridine, thymidine. Okay, here what are these nucleosides? Nucleoside is a combination of the pentose sugar and nitrogen base. Pentose sugar and a nitrogen base. As this is a question related to DNA molecule, the type of pentose sugar that is present is deoxyribose sugar. Here the molecular formula is C5H10O4. This is a molecular formula of deoxyribose sugar present in DNA molecule. Apart from that one, they contains nitrogen base. Here how many types of nitrogen bases are there in DNA molecules? DNA contains 4 types of nitrogen bases. Out of them we know that 2 are purines and 2 are pyrimidines. Purines are adenine and guanine. They are represented as capital A and capital G. Then pyrimidines are cytosine, represented with capital C, and thymine, which is represented as capital T. So these are the 4 types of nitrogen bases which are present in DNA molecule. This deoxyribose sugar along with adenine will form adenosine. Then this is first type. Then deoxyribose sugar plus guanine will form guanosine. Third one, deoxyribose sugar with cytosine will form cytidine. Fourth one, deoxyribose sugar plus thymine will form thymidine. So these are the 4 types of nucleosides which are present in a DNA molecule. So from the given options, let us see which of the following are exactly represented that are present in a nucleoside of DNA molecule. If you see the option number 2, adenosine, guanosine, cytidine and thymidine have been given there. So question number 17, the correct answer is 2. Question number 18, all biomacromolecules have greater than 1000 Daltons weight except options: 1 proteins, 2 nucleic acids, 3 polysaccharides, 4 lipids. Okay, what are these biomacromolecules? Biomacromolecules are the molecules which are having molecular weight which is highest molecular, we can say that it is more than 10,000 Daltons. Here in the given options, that is proteins, nucleic acids, polysaccharides and lipids. Okay, out of all the 4, all biomacromolecules have greater than 1000 Daltons, but one of them is having less than 1000 Daltons, that is lipids. Despite of having less than 1000 Daltons of molecular weight, why this lipid is considered as the biomacromolecule means because lipids are present in the cell membrane, that is plasma membrane, and these are not present as independent units. These are in the combination and they are part of the cell membrane, and when the cell membrane is broken, so they are separated in the form of vesicles, and when they are separated as a form of vesicles, they are the part of acid insoluble fraction along with the other biomacromolecules like proteins, nucleic acids and polysaccharides. So that is the reason why, despite of having less than 1000 Daltons of molecular weight, lipids are considered as a biomacromolecules. Question number 18, the correct answer is 4. Question number 19, the most abundant protein in the whole of the biosphere is 1. Collagen, 2. Insulin, 3. Rubisco, 4. Trypsin. Okay. In the options, all the 4 are a type of protein molecules. Collagen is present in animals. Collagen is the most abundant protein present in the animal cell, but not in the entire biosphere. Then insulin is an hormone, so that is present in animals. And Rubisco is a one which is a most important enzyme of photosynthesis. This is an enzyme which is helping in the first reaction of C3 cycle. So it is having both the properties, carboxylation property and the oxygenation property. And this is an enzyme which is considered to be the most abundant enzyme present in the entire biosphere, and it is the part of near about 25 to 50% of soluble proteins present in a leaf of a plant body. Then fourth option is trypsin. It's an normal enzyme which is useful in the protein parts. Okay, from all the options that have been given here, we can consider Rubisco is the most abundant protein which is present in the entire biosphere. Question number 19, the correct answer is 3.